Hello, my name is Van Wu. I'm the research fellow in the University of Canberra. So I'm leading the cancer project in terms of identify the metastatic cancer cells in the human cancers. We use the blood to enrich the cells that we are going to use for the DAP-array running to isolate the circulating tumor cells and cancer stem cells. When we start, we use the blood and we label the blood with the antibodies to remove the non-cancer cells. By doing that, we can enrich lots of cells to be injected into the cartridge to be isolated as circulating tumor cells and cancer cells. Using the blood, we can detect the number of circulating tumor cells and isolate the cells for the downstream DNA and RNA profiles to identify the key proteins and key DNA RNAs involved in the circulating tumor cells and metastatic cancer process. In this patient, from 7.5 ml blood, we detected and isolated only one circulating tumor cells with the marker of expression CD45 negative and cytokeratin positive. We also added the proteins that we are interested into this panel, such as PD-L1 and EpCAM. As you can see, they are all presented, so we are 100% sure this cell is circulating tumor cells. In this patient, from 7.5 ml blood, we detected 2,088 circulating tumor cells in this panel. We further confirmed the circulating tumor cells by looking at their morphology. For example, in these CTCs, we can see the nuclei stand with blue color, with CD45 negative is not presented, with cytokeratin here is green. With standing with vimentin as our actual protein as a marker, we further confirm these CTCs is present in this patient. When we go through all the other CTCs, we are very confident that the circulating tumor cells we are isolating are the real CTCs. Using DAP-array, we can isolate circulating tumor cells from blood as well as cancer stem cells from fixed FFP tissue from the patient. So from the 10 microliter of samples we isolated, we can use this sample to be further analyzed into NanoString. This is the chip that we prepared using the robot. Now I'm going to do the analysis. By using this instrument, we can get the data in 3 months from the work previously done by a PhD student in last 4 years. From the isolated circulating tumor cells, we are making cover slips to be examined by 3D images. This has been done by Robert McCuaig.
My name is Robert McCuaig. I'm a researcher in Sudha Rao's cancer lab. Now what I do is, when Fan provides me with the isolated CTCs from the DAP-array, I take them and put them on a cover slip and label the proteins of interest so that we can visualize them in high resolution microscopy. These proteins characterize what we call the metastatic circulating tumor cells, so that we can look at our drug target of interest, which is LSD1, a marker for aggressive cancer, which is in the red, and that's CSV, or a target for immunotherapy, which is in the blue, and that's called PD-L1. And the green, of course, is our LSD1. What I'm able to do is take a 3D image, which is a composite of the entire cell, put together using the software and high-resolution imaging system of the Leica, a DMI-8 confocal microscope. And so we can get extremely high-resolution shots, as you can see. This is a very small cell, and we're able to get incredible resolution of the nucleus and how these proteins can also interact inside the nucleus as well, which is very important information. Now, what we can do is quantitate these proteins which characterize the aggressive nature of these tumor cells. And by quantitating them, we can track how aggressive a cancer is, how much of these proteins are being expressed, and how we can essentially target these proteins and treat them with drugs which knock them down. And essentially we can count large numbers of cells from just a single sample of blood, to get quantitative and statistical significance. And this can aid in both tracking the cancer progress in a patient and tracking the effects of our drugs on the cancers themselves.